presentations. Vibroacoustic signals of pellucidity, hemiptera coleorrhynca structures involved in their percussion and applications for the phylogenetics of hemiptera. Dr. Leonidas-Romanos.
Hello. Hi, you can hear me? Yeah. Well, very nice to see you all. It's an honor to start with the first two presentations of the conference. I hope you'll bear with me for the next half an hour or so. You might be wondering why I'm talking about pellucidids in a heteropteran-focused talk. But as I'm sure many of you know, the phylogenetic relationships of pellucidids, of coleorrhynca, are quite poorly understood. And many morphology-based classifications and molecular phylogeny support a grouping of pellucidids with Heteroptera, while many other phylogenomic studies and as well some morphological studies suggest that coleorrhynca are instead allied with the Auchenorrhynca. So their phylogeny is still unsettled, we can say. And my background during my PhD, I was studying bioacoustics. And I found that each suborder of Hemiptera is defined by a particular type of vibroacoustic organ. So, and of course, we still do not understand whether these vibroacoustic organs are homologous between suborders. But since they're so highly specific, I wanted to see whether studying the vibroacoustic device of pellucidids can tell us something about their phylogenetic position. So I'll just give you a bit of background on the other vibroacoustic devices of Hemiptera. So I'm sure most of you know cicadas. So cicadas and Auchenorrhynca as a whole have elastic recoil devices, which essentially means there's essentially a spring that stores energy, and upon release, it releases a huge amount of energy to produce a fast motion that is way faster than muscle action could do alone. And the most famous example in Hemiptera are the timbal organs. So externally, the timbals have rib-like structures that you can see, and they're bent by a powerful muscle, and then they're released and they buckle and they produce a loud pop, which is amplified through air sacs. So this is how they produce their songs. And in our previous research, we have shown that timbals define cicadomorphs as a whole, even in fossils. So I'm not sure how easy it is to see, but you can see the ribs in all the cicadomorphs, on the timbals of all cicadomorphs. So it's a really old mechanism, and it's found throughout cicadomorphs. And more recent research of ours found a new mechanism in fulguromorphs, the snapping organ. It works completely differently compared to timbal, but we're still not sure whether it's homologous with timbal organs or not. But it's also an elastic recoil device, and it's found throughout the fulguromorphs. So again, it's yet one more vibration-producing device that essentially defines a Hemipteran suborder. And closer to home, Heteroptera are less specialized for vibroacoustic production compared to Auchenorrhynca. So they do produce low-frequency vibrations by essentially just moving their abdomen up and down. And the way they do this is by having the first two abdominal tergites fused into what is known as the tergal plate, and they essentially move the abdomen up and down. So it's a less specialized mechanism compared to the Auchenorrhynca. So what about coleorrhynca? So the first reports came from Hoch in 2006, where they recorded vibrational signals in pellucidids, but the precise mechanism wasn't discovered. But then my colleague Viktor Hartung recorded many more species and essentially found that vibrational signals are produced by all species of pelluridines. But they're also similar to Heteroptera in that these low-frequency vibrations are between 100 to 500 hertz. And they don't seem to have any specialized device to produce, higher frequency signals. And they are probably just producing simple abdominal vibration by moving the abdomen up and down. I'll show you in this video. So it's a very slight motion. So it's just moving up and down, but we haven't found the precise way they do this biomechanically. So we wanted to study the internal and external anatomy of this organ, of this group in more detail. 1 sec. So we used micro-CT, scanning electron microscopy, and confocal microscopy to study the vibrational organs of Coleorrhyncha. And we didn't find any type of elastic recoil device, either externally or internally. Their abdomen is quite simple. Essentially, they have a tergal plate-like structure like Heteroptera, so the first two tergites are closely associated. It's these two parts here. And when we looked internally with micro-CT and reconstructed the muscle system, again, nothing really special came out. They're just like normal longitudinal muscles that do not seem to be modified for high-frequency signals. And when we also looked at the dorsoventral musculature, which is involved in sound production in cicadas, for example, again, we didn't find any particular specialization in pelluridines, but we did find that the arrangement of the dorsoventral muscles is in doublets and triplets, and this conformation is actually extremely similar to the conformation that you find across Heteroptera. So I'll talk about that later, but it is quite interesting how, at least superficially, similar they are. And this arrangement is not found in Aucanorrhyncha. And we also examined some old fossils of related families, so Carabacidae, it's an extinct family of Coleorrhyncha. And we found that the morphology looks very, very similar to the extant species. So I have a direct comparison here. So you can see that the first segment is like sort of triangular, and the second segment is like a strip. So it's a very conserved device. And we thought that, okay, since Hemiptera seem to have very deeply conserved and highly specific vibroacoustic devices, why don't we try using these characters to plot them in a phylogeny and see what they can tell us about the position of Coleorrhyncha. And we did that using maximum parsimony in TNT, using 18 characters. So we did two approaches. The phylogeny on the left is using only the vibroacoustic characters, and we essentially find that Heteroptera are sister to Coleorrhyncha. But when we add those vibroacoustic characters to 93 morphological characters used by Viktor's previous phylogenies, we recover the relationship with Aucanorrhyncha. So it's quite an opposing, quite opposite results, really. So we are still, this study is still on the way. So we would like to hear also your opinions about interpreting these results as well. So we find that pelluridines have very simple and non-specialized vibrational signals, which seems to be similar to Heteroptera, but we cannot say for sure, is this a symphysiomorphy shared between Heteroptera and pelluridines, or is it that pelluridines had a more complex mechanism similar to Aucanorrhyncha that then got secondarily simplified? So we're still not sure about this. Also about the two phylogenies, one of them uses vibroacoustic characters only. Could it be that these characters are more informative about phylogeny than others, let's say? We're still not sure exactly. We're still not sure. And obviously, we would like to examine more ancient families of Coleorrhyncha to see what conformation they have. It's just most of them are compressional fossils, usually just a wing, so we haven't really found a good fossil where you can study the abdomen. And of course, we have absolutely no idea the genes responsible for the formation of vibroacoustic organs in hemiptera. So obviously this is quite a big knowledge gap, and it would be hugely informative on this debate. And from a bioacoustic perspective, we show that essentially if one compares vibroacoustic signals across hemiptera, it seems that simple abdominal vibration was the first step, and then elastic recoil devices evolved secondarily, which sort of makes sense. But still there's a huge knowledge gap for many of these vibroacoustic organs, and although we have studied the morphology of the pelluridid organ, we're still not exactly sure how it works. So thank you very much, and I'll take some questions. Any questions?
Any questions?
Yes. Christian?
Okay, really cool talk. Thank you very much. So my question is, why you characterize the abdominal vibration as a symplasiomorphy? Because really, I think what it might be, if it's homologous, right, across the groups you describe, it actually would be a synapomorphy of hemiptera that then gets, sort of transitions into something else within the two parts of Ochorrhyncha, I guess. Yeah. But it's really cool, actually, that it seems like it may be a synapomorphy of all of hemiptera, if it's homologous.
Yeah, I agree. I think synapomorphy is probably the best term to describe it. And yeah, if one studies some Sternorhyncha and also Heteroptera, and now we have pelluridid, it really does seem that vibrational signaling was present at the root of hemiptera. It's just a really big question is, are these organs homologous with each other? So it's still unresolved. Thank you.
Thanks, Leon. That was great. I just have a general question. So what's the context in which they actually do the vibration? And if it's in sexual situations, are you seeing any dimorphism between the males and females? Or is it they both got the same structures?
Yeah, so you mean only for pelloridids?
Yeah, let's go for that. Yep.
So, so Viktor collected that data. He would be best to answer. It's usually used, I think so far it's used only in a sexual context. But Viktor, maybe would you like to answer? Yeah.
I just wait for the mic.
Viktor.
Thank you. Sorry. Can I put it in? Thank you very much. Sorry. Yes, first the sexual context. The females do not sing, so it's only males, and it also depends on the species. I had this, what Leonidas showed, that was this South American species. They were vibrating like crazy. They were not disturbed by anything. And this scene that you sing, I think that was even 3 males. So male sitting on a male and another dead male hanging from a tree. And they were like riding on each other and doing this. So I once saw a scene of 3 insects. One below was female, then a male above it, and then a male above it, and the 2 males above were vibrating. So they were vibrating like in many contexts. As for New Zealand species that I recorded, they were very easy to disturb. I remember me seeing them in the acoustic chamber, like I see that the records is right, and I see that they're doing something, and I approach, and they probably notice my steps, and they stop. And but still, they would also just sing on alone. When they let them, I sometimes recorded them overnight, and during the night, it's like at 11, they would start vibrating and singing just sometimes on their own, alone, sitting all alone. So it's definitely, it has a sexual dimorphic component, but the context I still have to process all this month of recording that I have, so maybe I found some more concrete things, but it still it still seems that there are many contexts in which they sing. If you grab them, I think they will stop. The ones that are disturbed by my steps, they will stop definitely. The ones that are less disturbed, maybe I didn't try to grab them, but I think that they're also quite small, like 2-3 millimeters. I don't think you can hold them in a way that they still vibrate while you hold them. I think it's complicated.
Sexual selection and predation drive the repeat evolution of stridulation and atrophy. Leonidas-Romanos.
What is the... Well, yeah, me again. So it's on a related topic, as you can see. So just a little bit of background. We are all familiar with acoustic signals that they're used by many arthropods. But a fairly recently discovered mode of communication, which seems to be the dominant ones in arthropods, are the substrate-borne vibrations, so vibrations that pass through materials and do not require a tympanum to be perceived, so they're not airborne, just vibrational one. So this seems to be the biggest player in hemipteran communication as well, and I'll be talking about this. So, well, as I introduced before, there are 3 or 4 types of hemipteran vibroacoustic organs. So yeah, timbal, snapping organs, tergal plate, and the pylorideid device, which may or may not be similar to the tergal plate. And these are all abdominal-based mechanisms. However, as I'm sure you all know, many Heteroptera use stridulatory devices, which are very well known in Heteroptera in terms of, most of us will be familiar with them, and they're frequency amplifiers in the sense that they allow the insects to produce signals that they would never be able to produce by muscle action alone. And they're developmentally very simple to produce, so it's essentially just a few teeth developed, let's say, on a leg that strike against the abdomen on a modified surface. And here you have the typical Sidney mechanism, so some striae on the abdomen that rub against teeth on the wing. And most of us probably know that there are many types of stridulatory mechanisms at Heteroptera, but how many exactly? This has been quite a difficult question to answer, because they really do have a vast number of stridulatory mechanisms. And although they have been sometimes cataloged at the genus level or sometimes for the infraorder level, we do not yet know how many stridulatory mechanisms exist throughout Heteroptera. We also do not know how many times have evolved, and we are also not sure what they're used for behaviorally and how they appeared, and whether the Heteroptera are able to hear the sounds that they produce with the stridulatory mechanisms. So the goal of this research was to answer all these questions. So I essentially read a lot of papers, I think 30,000 to be precise. It took 9 years. So now I think we have a good understanding of how many stridulatory mechanisms exist in Heteroptera, and I mapped them on their phylogeny. And then to see how many times they evolved, I wanted to understand their homologies. And in most cases, it was fairly straightforward to see whether a structure is not homologous. So for example, a stridulatory mechanism on the abdomen and the leg is probably not homologous with a structure involving, let's say, the head and the leg rubbing against each other. So it was fairly obvious. But in the cases where fairly closely related taxa use very similar mechanisms, I used the maximum parsimony following the doublet rule of Madison et al., which I would summarize in this figure. So if the taxa on the left of that tree have the same mechanism with the ones that are on the far right, since they're separated by multiple lineages that do not stridulate, the most parsimonious explanation is to say that the top left and top right lineages evolved these mechanisms independently. So some of the results of this study, I found that Heteroptera have 25 different types of stridulatory mechanism, which is actually quite impressive, as in Coleoptera you only have 14, and they're quite well studied, so quite impressive. However, from all of these mechanisms, 3 of them account for 82% of all the independently evolved mechanisms. So there's a huge morphological bias. And the 3 primary mechanisms are essentially the tergal plate rubbing against the wing, so the Sidney mechanism that I showed before. The other one is the leg rubbing against modified surfaces on the wing, and the abdomen, the leg also rubbing on modified surfaces on the abdomen. And I will explain later why this bias may arise. But overall, as you can see, I plotted all the plectra and the stridulatra that can be found in Heteroptera, and you can see that virtually every body surface is exploited. Obviously, this is not one species, this is across Heteroptera. When we mapped the stridulatory organs on their phylogeny, we found that they've evolved 84 to 98 times independently, which is quite a staggering number. And as we examined all other arthropods as well, there's actually no parallel among animals for the independent evolutions of this character. Just to explain the discrepancy between 84 and 98, there are some stridulatory organs that have been described as stridulatory, but we're not really sure if they really function as such, so they are known only from preserved specimens. So in one phylogenetic estimate, we excluded them, and you get the 84 numbers. And in a more relaxed estimate, we included them, and you get 98 times independently. And the image on the right is a bit complicated, but the most important thing is to see that the red bar shows what percentage of each infraorder stridulates, and I have summarized them here. Dipsocoromorpha were not included because they do not have any confirmed stridulatory devices. And you can see that Simicomorpha and Nepomorpha are the absolute champions of stridulation, and we can discuss about this later as well. So why do Heteroptera stridulate? So when we plotted the known behavioral context, it seems that defense and behavior account for 60% of the behavioral context where stridulation is used, and all other contexts also involve sex or defense in some way. So for example, they use competition, they used in competition, but also defense, etc. But we should also be aware that there could be some bias in these results, because for 79% of stridulatory Heteroptera, we have absolutely no idea why they use it for. So I think an orthopterist once said that the entomologist's ethological observation start from the net and end in the killing tube. So this is certainly the case with Heteroptera, unfortunately. But when I compared the results of Heteroptera with all other arthropods, you essentially find the same pattern, and you actually get an even stronger bias towards defense. So sex and defense does really seem to be the key driver of stridulation. And I just wanted to also show that, first, you can see that Heteroptera really are the absolute champions of stridulation. Only arachnids are comparable. So they even surpassed Orthoptera, which are like the other most renowned singing insects. And also we found that the three dominant stridulatory mechanisms Heteroptera also evolve very, very frequently in other insects as well. And so why is stridulation effective against predation? So it seems that some predators it seems that against some predators, stridulation is quite an effective strategy, in others not so much. And we're not sure why there is this discrepancy, but in the predators that it is successful, there are several theories about it. So it could be that they just get startled by a sudden acoustic signal. It could also be part of a bluff, like a deimatic display. It could also warn of venom or noxious chemicals. So reduvids, almost all reduvids stridulate, and they can also give you a painful sting. So it could be advertising a noxious property, and it could be part of Mullerian mimicry. So some largids look very similar to velvet ants, and they also stridulate, and velvet ants also stridulate, so it could be Mullerian mimicry involved. And regarding the use of stridulation in a sexual context, its adaptive significance is slightly less understood, but there are some evolutionary biology theories that suggest that females will select over time for more complex sexual signals. And given that most hemiptera only have abdominal vibration as a means of producing vibroacoustic signals, stridulation could also be an extra supplementary way of stimulating the female in courtship, for example. But of course, another interesting question would be which came first? Did stridulation first appear as a defensive signal and then became co-opted as a sexual signal, or vice versa? And probably either could be the case for different lineages of Heteroptera. And you might wonder, why are 3 stridulatory devices the most common in Heteroptera? What we suggest is that because that this is the case because they arise from a similar behavioral background. So many ethological observations across Heteroptera show that prior to mating, many Heteropteran species perform some type of cleaning motions when they're facing each other. And what these cleaning motions involve is usually the leg cleaning the wing margin or the abdomen. So you can easily imagine that the next stage of this behavior is to modify the surfaces and produce an acoustic signal. And also using the tergal plate, they also many times rub the wings. So it could also account for why the stridulatory tergal plate has arisen many times. And one of the other key questions is whether the Heteroptera are able to perceive the acoustic component of the signals that they produced. And it seems that it's not really the case. So most Heteroptera lack tympana, and most ethological studies have shown that they are absolutely deaf to the acoustic component of their signals, and they only perceive the substrate-borne vibrations of this component. And this would make sense if most of these acoustic signals are used against predators. So it's for interspecific listeners, so there's not much point for Heteroptera to listen to their own signals. Obviously, in a sexual context, this changes. And only nepomorphs are able to perceive their acoustic signals because they have tympana. But one could be tempted to say, okay, so they probably evolved tympana in order to hear their songs. But since not all nepomorpha have stridulatory organs, the more likely explanation is that tympana evolved for some other purpose and then became co-opted to perceive acoustic signals. So to summarize, now we do have an atlas of the stridulatory mechanisms across Heteroptera and most other arthropod groups. So this will allow us to essentially target some mechanisms for behavioral studies and biomechanical studies. And we are the first to show that, that sexual selection and predation are the predominant drivers of stridulation. And we also show that there is a lot of morphological bias as to which body parts are more prone to evolve for stridulations. And unlike many other groups, for example, tetrapods, which you see a clear-cut sensory and signal coevolution, this doesn't seem to be the case in Heteroptera, and this is also the case with other groups of animals. For example, some moths cannot hear the stridulatory signals they produce because they're again used against predators, and snakes, some snakes also stridulate, and they're obviously deaf. So it's quite an interesting addition to this pattern. So thank you very much for your attention, and looking forward to your questions. Thank you.
Any questions?
Toby. Yes. Okay.
So you mentioned. Thank you. Thank you for the talk. You mentioned that the Nepomorpha are champions of stridulation. What context do they do it? Is it within the water, or did they come out of the water to generate this? So how does that work with the three-dimensional environment where they live?
Yeah. I think almost all Nepomorphs, apart from some Gelastocorids, stridulate underwater. Gelastocorids live on the shore of rivers and stationary water. But yeah, most of them do it underwater, essentially. Yeah.
What's the effect of that? Stridulation, usually you would expect some sort of sound to come out.
Yeah, yeah.
That is going to interfere with or is going to be disrupted by the water.
Well,
so is it just vibrations creating ripples or something like that?
No,
or
no,
is
it's acoustic signals, and some of them are among the loudest insects on the planet. So micronectids can be heard from many, many meters away, away from a pond. So it's transmitted both inside the water and outside sometimes. So it's quite a powerful signal. And it seems that water is quite a good conducting surface for acoustic signals. In geromorphs, very, very few of them use stridulatory signals, and they're obviously on the water surface, and we're not really sure what they use them for. I think we do not know the behavioral context in any of them.
I do have a question about rediviots.
Yes.
So, because rediviots would stridulate in the context of defense as well as mating.
Exactly.
Right? So, do you have any suspicion as to how they can hear their partner?
Yeah, so when they stridulate, they generate both substrate-borne vibrations and acoustic ones, and the reduvids almost certainly just respond to the substrate-borne component. Yeah.
Did you have a look at whether there is some connection between the or so thought anti-predatory function of the stridulation and the size of the bugs that produce it, in terms of that larger size bugs like reduvids are more prone to being predated by birds, which are probably going to be affected by the startling of the stridulation, and whether there is some connection that the smaller bugs tend to have rather sexual or non-stridulation and the larger ones tend to have the anti-predatory ones.
Actually, that's a very interesting question. I haven't examined that, but I can tell you that well, certainly very small heteropterans can stridulate, so many tiny cydnids stridulate, many small reduvids do the same. But many choraids, for example, stridulate. They're quite big animals. I'm not sure. It would be interesting to see it statistically, but actually, that's a very good topic for a paper, I would say. So thank you.
And one more question about the nepomorphs stridulating. is it known to be like some form of anti-predatory, or is it like in case of nepomorphs mostly sexual?
So yeah, the nepomorphs are the best studied heteropterans in terms of the behavioral context of stridulation, and they use it for everything, really. Competition between males, so territoriality, sex, defense, yeah. So yeah, it's quite broadly used, actually.
Have you looked at the ripple communication? It's not by sound. In one species of jarrets, the males will tap on the water, and this is a sexual a signal to get the females to come. It has only been done on one species, has never been reported in other species, so that might be interesting. So instead of using tympani to detect the sound, the ripple, it will be probably some organs on the feet.
Yes.
That might be interesting to look at.
Yes, exactly. So it seems that vibrational signals
Yes.
were present at the root of Heteroptera, and almost all Heteroptera that I've examined used vibrational signals, so abdomen-based, but also, as you say, jarrets also use ripples for their communication.
Also, there are some jarrets, but this was done in Rogovilla, I think.
In Rogovilla?
Rogovilla, right.
Yeah, and Jarus as well. Aquarius has also been studied for the ripples, and also actually some nepomorphs also use ripples, abdomen-based ripples to communicate.
So they are detected rather than heard.
Exactly, yeah, they're detected with the general acoustic, well, substrate-borne vibrational sensors on the
Yes.
leg, so the chordotonal organs.
Thank
Yeah.
you.
Okay, I've got a question. Have you looked at the likelihood of animals that have behavior versus diurnal behavior actually more likely having stridulation or not?
Oh, that's actually quite...
Sorry, could you continue a discussion and coffee?
Sorry?
Time finish.
Oh,
Okay, just very briefly. Well, I haven't looked at it, but we can discuss it. It's quite interesting. Thank you. Yeah, thank you.
Thank you, sorry.
¿Quién toca ahora? ¿Tatarnic? Vale, voy. Dame un segundo.
Working? Hello?
Hi there. Hi there, can you hear us?
I can hear you. Can you hear me?
Yeah.
Hello.
All right, we just need to be able to share a screen and then I can share the talk, I guess.
They're going to present you right now and go forward after the presentation.
Okay.
The fucking channel, morphology of Heteropteran genitalia elucidated for its ray microtograph, Nikolai Tatarnic.
All right, you need to give me screen sharing. So I think if you make me a host or something like that, then that enables the screen sharing.
Apparently,
Oh, there we go.
I've allowed
There it
you to
is.
share. Yeah.
Excellent. Okay, so let me start this up.
Just in case.
All right, is that showing?
Yep.
And are we live? Are people there? Can you hear me? That sounds like a yes. Okay, good. Sorry for the, I wish I was there. Circumstances didn't allow me to be there, so my apologies. And so today I'm just going to give you a little bit of a talk about the work I've been doing using micro CT on heteropteran genitals. All right, so this is, I guess I'll state the obvious. Insects are really small. The genitalia are really, really small. So there's always a challenge in imaging reproductive organs in insects. Nonetheless, people have done some amazing stuff with light microscopy, histology, confocal, all that. There are limitations to every technology. So there's limitations to depth of field and distortion of images, and also, of course, destruction of specimens. So that's one of the reasons why I got into this micro CT business. So micro CT, it's a lot like a CAT scan, in the health professions, except that it's really small. So it's used for small objects. Basically, an X-ray beam is passed through a sample, and then the X-rays get absorbed differently by different tissues. So the X-rays that pass through it differentially, so you get a grayscale of absorption, basically. The specimen is rotated through 360 degrees, and then you get a whole bunch of images. These are all stacked together into what looks like a flip book. You can flip through them through a sagittal section or a cross section. And then this is later compiled into three-dimensional volume or point cloud, which you can then analyze and play with and make videos and make images. So the machine I've been using, it's an XRM, which is an extension of X-ray of micro CT. So normally, X-ray magnification comes just from geometric magnification. So the closer the source is to the object, the greater the magnification. The farther the detector is, the greater the magnification. But XRM also adds in a scintillator, which converts the X-rays into visible light, and then that's passed through a lens, so you can get even more magnification. So the system I use is at the University of Western Australia. It's a Zeiss X-Radia 520. And this machine can, I think the resolution gets down to about 0.7 micrometers for a voxel. So a voxel is like a 3D pixel. And in this image, you can see on the left-hand side, it just looks like a big box. Inside it, there's the source on the left, the detector's on the right. And if you look really close, that's my sample. So this is how I typically scan my insects. They're almost always scanned in ethanol, although sometimes I'll scan pointed specimens. So in here, it looks like I've got two probably pygophores of I don't know, rudiviids or leaf-footed bugs or something. And you can see the detector and the scan and the source are very, very close to it to try and maximize the image. All right, how did this all start? Well, it started with my interest in Corodromius, the traumatically inseminating plant bugs. And in this image, this is just to show you that with X-ray data sets, you can actually separate out, you can tag and remove different tissues or different organs if you want to work on them separately. So here I've tagged the pygophore, the Adyagus, the left paramere, the right paramere, and I've separated them. But you can also look at them all together. And of course, just as a reminder, it's when you scan the whole animal, you're not just getting these solid structures, you're also getting the soft tissue inside. So this is a cross-section of an undescribed species of Corodromius. And you can just quickly, you can see the eggs, you can see the flight muscles here. And then this big thing here, this is the spermoledge. So that's that immune organ that deals with the sperm and stuff when it gets injected into the side of the female. All right, let's see if this works. I know I was told not to do videos, but I'm doing a video anyway. This is just to show once you have the separate pieces, the separate volumes of your data set, you can actually animate them and rejoin them together. So this just shows how this is Corodromius nakatanii, and this just shows how the pygophore and the right paramere act to brace the left paramere while it's used for stabbing into the side of the female. So you can see how the right paramere acts like a little hand. It's got a little thumb on it. It grabs in. All right, so I've also been using using this CT technology for generating plates for taxonomy. So in this case, I had a species that I was working on with Jerry Cassis, and we had very few males. So I ended up dissecting it. In the end, I find you still have to dissect the specimens. You still have to do the work. It just makes for easier imaging once you've dissected it than having to draw it under camera lucida or photograph it. You get more depth of field. So this was a straight up taxonomy paper, but of course, it gave me all this data that I can still use for other things. So Jerry and I are actually working right now on looking at the internal anatomy of chorea genitalia. So I've scanned several other ADA that have been inflated, and yeah, you can see all the structures inside. You can see what's going on. So it's helping us answer some questions that we've had and as to what goes where and what parts are affiliated with what. And also this data is shareable. You can share it as PDF files, as videos, as still images. You can redissect it in any direction you want to, and it's a permanent record. So even if your dissection goes missing or you used KOH and it eventually dissolves into nothing, at least you'll have these nice data sets. The next example, I just want to talk a little bit about Veliids. So this is a project on that I did with an honor student, Paige Maroney, and her co-supervisor, Kate Bryant, at Murdoch University. And this project stemmed from reading some Anderson and Weir work where they revised the genus Ostromicrovelia. And in Ostromicrovelia, the males and females both have really crazy elaborate structures on the abdomen. Jerry Cassis and I had a previous honors student, Mitch Hodgins, and he worked out a phylogeny of this group. We've since sunk them into the genus Nesidovelia, and we proposed that sexual conflict drives these dimorphic structures. So yeah, so I got Paige to help me with taking this to the next level. So just as a little bit of background, in water striders in particular, sexual conflict is thought to be one of the main drivers of their mating system. So essentially, males and females have different reproductive optima, and because of that, there's a conflict over when to mate, how often to mate, and so on. And this can lead to grasping structures in males and anti-grasping structures in females. And so here's some pictures of Nesidovelia, the ventral surface of the abdomen of males. The most striking thing you'll notice is on segment 8 of the male, just before the pyodophore, they have these crazy processes sticking out, and these often are matched with structures on the previous segment, on segment 7, and it looks like they articulate to form a clamp. So we thought, okay, well, this is a clamp, but what does it clamp onto? And in the females, they have the connexiva are convergent and form a keel on the back, and there's tufts and setae and prongs sticking out of them. And also their genitalia is recessed into the body and is hidden behind a drawbridge-like proctoger. So it's behind a shut door, basically. All right, so what we did, Paige sat for hours and watched bugs mating and then tried to freeze them with liquid nitrogen. In the end, we managed to freeze 3 pairs in copula. And then these were then slowly thawed out and then glued onto a point and then kept in ethanol. So these things are all kept in liquid to keep all the organs more or less where they should be. And just to show you, this is the posterior ends of the males and the females. So on the left, you've got the female. You can see in pink is the, or red or whatever color that is. That's the proctoger blocking the female genitalia. And it's biconcave on the underside. And the male has this process with 2 prongs on it on segment 8. Now, if we expand this part of the abdomen here, so from this scan, you just expand that and cut it open, you can see that the male segment 8 process grasps onto the underside of the proctoger, pries it open and holds it against his segment 7, and then the adiagus is inserted fully into the female. And I don't know if you can see on that image, but at the tip of the male adiagus, you can see a little skinny tube coming out. That's the ductus of the male, and that's actually going into a big coiled spermatheca of the female. And this, again, it's another video, sorry. This is just to show you, so we're continuing with this work. I'm looking at the other species in the genus because there's a lot of different variations on this proctoger and male clamp. So this just shows once you've got these X-rays, once you've segmented them out, you can actually articulate them and animate them into movies. So there you can see there's the, this is tuberculata, that's the male's clamp. And then there's the female's proctoger, which is pretty simple. There's no modifications. In Distincta, they have this long, skinny letter opener like thing sticking out of their segmentate process. And we're not, I don't think that it doesn't look like the kind of struct that has much strength behind it, if you know what I mean. So it doesn't look like it's something that's jimmying the female open or forcing her open. But if you look at the female, she has on her on the posterior margin of the abdomen, there's actually these little grooves. I don't know if I can point with my mouse. Yeah, under here, there's all these little tiny grooves, and I think that little letter opener slides into one of these. And I suspect because a lot of these species live, there's multiple species living together, so I think this might actually be more of a species recognition structure than anything else, because these things, their mating system, it's basically the male jumps on a female without any previous assessment. So this probably helps to limit interspecific mating, although that has to be tested. All right, and the last example I want to talk about is Haltocotoma. So this is a project that was started by Katrina Menard, and I'm working with her and Michael Schwartz. So these are plant bugs in the tribe Acridotarsiani, and they live on yucca, hence the name yucca plant bug. They have really cool genitalia, and another cool thing about them is that when they die when they're mating, they sometimes stay in cop. So Katrina sent me several specimens that were in copula, and we scanned them to see what was going on the inside. So just to orient you around their anatomy, here's a pydrophore of Nikolai, which is a different species, but pretty close anyway. So here's the left paramere, there's the right paramere, there's a process on the pydrophore, and then there's the adiagus running through the middle. And some dissections from Michael Schwartz. Thank you, Michael. This is the female. You can see the seminal depository big bag at the top. The ventral labiate plate. This is, I think, what Katrina was originally interested in, is these crazy teeth on this structure, because it looks like a bear trap, sort of. And you can barely see in this image the lateral oviduct there, but they are there, trust me. Now here on the left is an X-ray, and on the right is a dissection. You can see they're pretty comparable. And you can see the same structures. There's a capitate processes, there's the ductus seminis. And if you look closely at the ductus at the tip, it's sort of sharpened almost like a spine, but just before the end of the spine, there's an opening. So this is just a magnification of the scan to show that there's this little sort of slit-like opening there. And here, this is to orient you around the scans. This is, imagine that there's more bug attached to these scans. I've reduced the scan just to the business end of both animals because the data set is too big otherwise. So here you can see that's the ovipositor of the female. There's the pydrophore of the male. That's the left paramere of the male. And this is a ventral view of the same thing. Just to orient you, there's the coxa of the hind legs. There's the left paramere. There's the process in the pydrophore. So the male uses that left paramere and that process to pinch the sides of the female abdomen. And the female abdomen is sort of hinged and it articulates. So it bends about 40 degrees sideways. It's really weird looking. There's the right paramere, and then that's grabbing onto the adiagus, so holding it in place as it's fed into the female. Okay, now I'm going to walk you through some still images. This is from a video that I did just a few days ago, but I wasn't sure if the video was going to play. So this is just some stills to show you. So we've got the female and the male. Start looking at it from the back, slicing into it, slicing in closer. And now if you look really closely, you can see this is the beginning of the male's ductus, or rather the ending of the male's ductus. You can see it becoming visible. And we slice in a little closer and a little closer. Now, if you look right here, you can see these are the lateral oviducts. So the ductus is coming in right below the oviducts. And if you look a little bit closer, you can see there's the secondary gonopore. So there is where the male's ejecting his sperm right under the oviducts. And then if you look at it from above and clear it out a bit, you can see this is the dorsal sac of the female genitalia. And that's nowhere near the seminal depository. So we probably need to rename some of these bits and pieces. Alternatively, other mirids might be putting their sperm in different places, but at least in this case, it looks like they're putting it near the oviducts. So just to recap what's going on in here, right paramere braces the aegagus, left paramere pinches the female abdomen, sclerates on the dorsal surface of the ventral labiate plate act to guide the adyagal guide. I didn't mention that before, but that's what's going on. The ductus extends all the way caudally into the dorsal sac, and the secondary gonopore opens below the lateral oviducts. All right, I just want to thank Katrina and Michael and Jeremy Shaw at the UWA, who's helped me with learning how to use their CT machine, Paige Maroney and Kate Bryan, and of course Jerry Cassis, who is always involved with my work. And if anybody has any insects in copula or wants to see what's going on inside them, I've managed to score a pretty sweet deal on this machine. So I've got hundreds of hours of scanning time. So if people want to look at their insides of their bugs or look at genitalia, I'm happy to oblige. And just lastly, if this works, I just will show you this video just because it's fun. Playing. There we go. All right, and that's it. Thank you very much. I don't know what happens now. So I'm not sure if there's questions or if that's a possibility, or is this, do I just go away? Okay, cool.
Hello. Thank you for this very nice talk. I have a question about the Nisidivilia, the males with processes. Do you observe hybrids at all in the field? And if you do, how do they look like and at what frequency?
All right, so could you type, could someone type that in or something? I can barely hear it. The sound sounds like a public washroom.
Nisidivilia. Yes.
Yeah. Yeah. So I haven't observed any hybridization, so I don't know if there are any hybrids. I just know that there are a lot of species that are sympatric, and there's a lot of divergence in the male and female structures. So it's just a working theory that that there's reproductive error that's been tested at all. But having watched them mate, there's no prior assessment by the males. They seem to just jump on anyone who goes by, and then the females go through this elaborate struggle, much like in Jared's, where they flip the male over and they try to flip him onto his back and do a bunch of somersaults and stuff. So the mating, and I've watched about 4 or 5 species, and it seems quite similar, the behavior. So it's just conjecture. You know, I don't know actually if they do hybridize, but I would suspect that it would be detrimental. So you'd expect some sort of reproductive barriers.
Thank you.
No worries.
Okay. Thank you very much. There's no more questions apparently.
Right, well, thank you very much, and enjoy the rest of the conference.